A Day in My Life as a UP Manila student.
First year, second sem Animal Stimulus Edition.
Hello! Today, we are exploring the world of animal taxis, the directed movement of an organism toward or away from a specific stimulus. Every living thing possesses irritability, which is the fundamental ability to detect and respond to changes in the environment. In this exercise, we are focusing on the Lumbricus terrestris, or the common earthworm. Because earthworms lack complex sight or hearing, according to Wilson et al., they rely heavily on specialized sensory receptors across their body segments to navigate their world.
Navigate!
By observing how they react to different physical and chemical triggers, we can better understand their survival strategies and physiological limits. Our objectives are: first, determine which regions of earthworm, the anterior, middle, or posterior, exhibit the highest sensitivity to external stimuli. Second, describe and predict whether an earthworm will display positive taxis or negative taxis when subjected to touch, light, heat, moisture, and chemicals. To start our exercise, we need our materials.
Siyempre, alangan naman tayo ang samples, di ba?
First is, of course, we need the earthworms.
Hoy, hindi yan yung earthworm.
Dito.
Nasa soil.
Next is our hand lens for creating our heat stimulus. The card with 2mm hole in the center. Together with the card, we need the flashlight to create our light stimulus. The xylene for our chemical stimulus. Dissecting pin for the touch. And the dissecting pan.
Siyempre, baka tumalon-talon yung uod natin.
Animal taxis. Ano? Tara? Tara na! Next are the methods.
In preparation, line a cleaning dissecting pan with a damp sheet of paper. Clean the active, live earthworm and place this on the paper.
Parang pwede nang iprito ah. Yummy!
With contact as the stimulus, lightly touch the different body regions using the point of dissecting needle. After 5 minutes, apply repeated stimulation to the middle portion only.
So kumbaga, parang kikilitiin nyo lang siya.
To test its reaction to light stimulus, use a card with small 2mm hole in the center and a flashlight. Allow a spot of light to shine on different regions of the worm.
Hawak mo ang light, hawak mo ang light.
On the other hand, to test its reaction to heat, with the hand lens, focus sunlight successively on different body regions of the earthworm.
Aray ko, ang init!
For the moisture, line a clean and dry dissecting pan with tissue paper. Dampen one half of the tissue with water and leave the other half dry. Place the worms at random over the entire surface. After an hour or more, observe the location of the worms.
Saan pupunta ang mga uod?
And lastly, to test its response to a chemical stimulus, moisten the tip of a glass rod or stir rod with xylene and bring it close to the different body regions of the worm.
Xylene ha, hindi asin.
Here's the summary of our results. Now for the discussion.
As observed, the earthworm moves away from the point of contact,
Worm was like, wag po, wag po, wag po, wag po.
demonstrating a negative response from the stimulus. This behavior is supported by the study of Matsukete et al., which explains that touch activates specific sensory neurons that triggers escape or reversal movement. The anterior region is the most sensitive, showing the fastest and strongest avoidance response by moving backward, while the posterior region moves forward in response to contact. The middle portion initially exhibited strong withdrawal movement by wriggling, which gradually decreases after continuous stimulation. According to Yang et al. And Bernal Gamboa et al., this reduction in response is explained by habituation, a process in which repeated stimulation decreases behavioral reaction over time.
According to Takeshi et al. In 2020, thermotaxis refers to an organism's movement toward or away from a heat source. In this experiment, the earthworm exhibited negative thermotaxis as it moved away from heat. This finding is supported by Pri and Sankarb in 2025, who identified heat as a major stress factor for earthworms, noting that high surface temperatures and dry soils are more limiting to them than cold or waterlogged conditions.
Ang init!
As explained by Ricci in 2018, Earthworms are adapted to cooler, moist underground environments, and exposure to heat or sunlight put them at risk of dehydration and death. To maintain homeostasis, they move toward cooler areas, especially since they breathe through their skin and require moisture to survive. Additionally, the prostomium is considered the most sensitive part of the earthworm, as it plays a key role in sensing the environment and helping the organism navigate through the soil, as per Nature Watch in 2014. As observed, earthworms are sensitive to light and tend to avoid it. According to Drews in 2004, this behavior is called phototaxis, which is the movement of an organism in response to light in its environment. In this case, earthworms show negative phototaxis since they move away from the light source. Based on Sustainable Directory in 2025, they do this to avoid drying out and to stay safe from predators. Additionally, while all parts of the earthworm can detect light, the anterior end appears to be the most sensitive. This is supported by Hess's 1925 study, which found similar results and explained that this region has a higher concentration of photoreceptors.
After exposing to dry and moist environment, the worms tend to transfer to the moist site. This response is called positive hydrotaxis, which is the movement towards moisture or humidity gradient, according to Uri et al. In 2020. Mullen in 2021 explained that worms are highly dependent to moisture because they breathe through their skin through the process of cutaneous respiration, which requires a thin layer of moisture to allow oxygen to dissolve and enter their bloodstream. Furthermore, it is observed that the anterior part of the worm responds most sensitively, and this is supported by the study of Dominguez and Edwards in 2011, which states that the fleshy lip-like lobe in the anterior part, called the prostomium, acts as the primary moisture detector for the earthworms.
Sa moisture lang tayo ah.
Okay po. For this part, we tested how an earthworm responds when exposed to chemicals, specifically the substance xylene applied to different parts of its body. As you can see, when xylene was applied to the anterior region, we observed a faster and more coordinated withdrawal response. This is because chemoreceptors are more concentrated near the anterior end, allowing the worm to detect chemicals more effectively. On the other hand, when the middle or posterior segments were exposed, the reactions were present but generally slower or less coordinated. To elaborate, earthworms are capable of chemotaxis, which means they move in response to chemical signals in their environment. In cases involving potentially harmful substances, they show negative chemotaxis, which means moving away from the source, according to Loureiro et al. In 2005. Overall, the anterior region plays a primary role in detecting chemicals, while the rest of the body supports movement and escape, demonstrating chemotaxis as a protective survival mechanism.
Thank you for watching. That's all for today. Bye!